before I start, I really would like to thank Edith, Alain, and Nicole Ledoirin for this really nice invitation. It's a great honor for me to be part of this day today, because I must say that I think François Jacob was really a role model for a lot of scientists of my generation, but also of her generation, I think. Not only because of his major scientific achievements, but also, I must say, via his books, and in particular this one, which allowed us to envision that not only was he a great scientist, but he also seemed to have a lot of humanity, which to me is equally important. And so today, I would like to share with you some work that we've done trying to understand how mouse oocytes are able to put their nuclei at their center, so differently from xenopus oocytes that we've heard this morning. So as Edith just said, mouse oocytes are very important cells, and as François Jacob just said, they are fascinating, at least to me, I must say. So these cells, indeed, after a series of asymmetric division and fertilization, will give rise to the embryo. And so they are very important for this, I think. And so in many cells, the nucleus and the nucleus position can act as a morphogenetic cue, providing both spatial and temporal information to the cell. So for example, in fish and yeast, the position of the nucleus will define where the septum will form, and this is very important for the cell to equally partition its cytoplasm and its DNA into the two daughter cells. In many oocytes, such as the Drosophila and also the Xenopus oocyte, the position of the nucleus inside this cell will actually define the future axis of the embryo. Here you see that the location of the nucleus will define the dorsal ventral axis of the future embryo. And in most cells, the position of the nucleus is actually defined via its connection to the canonical centrosome, which contain a pair of centrioles together with pericentriolar material. And this structure, in most cells, actually nucleate this array here of microtubules which grow and shrink, and allows the cells to actually measure the force exerted by its environment and actively position its nucleus at its geometric center. So in most cells, it is the canonical centrosomes that are very important to position the nucleus. However, most oocytes, and this is the case of Xenopus, but also mouse oocytes, they lose their canonical centrosome during oogenesis for a reason that is still, I must say, unclear. And so instead of having canonical centrosome presenting a pair of centrioles, they have acentriolar microtubule organizing center that are here depicted in red that surround the nuclear envelope. And despite not having canonical centrosomes, they are still able to position their nuclei perfectly in the center of these big cells. And the failure to do so actually correlates with a poor chance of success of what follows, which is the meiotic divisions. So, as I just said, most mouse oocytes are able to position their nuclei in their center. And here, the nucleus has been labeled with a nuclear probe, which will stain the nucleoplasm. So these nuclei are very big, as John Gurdon illustrated earlier. They are 30 micron wide in a cell which is 80 micron wide, so it's very, very big nuclei. And what you see here in gray in the middle is actually the nucleolus that is not stained by this probe. And so most oocytes have their nucleus in their center. However, it was observed by two PhD students from my lab that in oocytes coming from the forming 2 knockout strain, their nuclei are off-center. So forming 2 is a nucleator of microfilaments, and the forming 2 gene is an essential maternal gene because the only phenotype that this total knockout has is actually female sterility. So it's an important gene. And in oocytes which don't express formin 2, instead of having a nice cytoplasmic mesh of microfilaments in the cytoplasm that you see here in green, and also microfilaments that surround this big nucleus here, the cytoplasm is completely devoid of cytoplasmic microfilaments. And their nuclei are off-center. And so Maria Almonacid, a very talented postdoc in the lab, decided to determine whether the lack of nucleus centration in oocyte coming from the formin 2 knockout strain was a direct or indirect consequence of not expressing formin 2 during the whole process of oogenesis. So she did a simple experiment, which namely was to add back formin 2 into the formin 2 knockout oocyte and follow nucleus movement. And so what she could observe is that after re-expressing formin 2 into these oocytes, then the nucleus would be able to be re-centralized. This centralization event is not an artifact due to the microinjection, because if Maria only expresses the nuclear probe, you see that the nucleus stays off-center. So it undergoes extensive deformation because the nuclear envelope is actually extremely deformable. It has a lot of microtubules that are associated to it. But the nucleus can stay off-center for actually hours. And so by performing this, Maria had in hands a very robust assay to test for nucleus centration. So, because indeed, in all formin 2 knockout oocytes, the nucleus is off-center, and when she expresses formin 2 back into these oocytes, then all oocytes will position their nucleus back to the center, whether Maria can follow the whole process or not during the time course of her movies. And so she could then track the movement of the nucleus as a function of time, and in particular, she could track the motion of the nucleus centroid, and she could measure the velocity of the nucleus centroid as a function of time. And as you can appreciate here, at the beginning of the motion, the velocity is relatively high and will progressively drop as the nucleus becomes centered. And it takes about 5 hours for the nucleus to reach the center of this big cell. And then you see that the nucleus centroid undergoes basal oscillation, and we think that these basal oscillations are probably due to the fact that the nucleus is actually deformable. So it drifts a little bit around the center of the oocyte. In formin 2 knockout oocytes that don't re-express formin 2, the formin 2 protein, the nucleus stays off-center, and you see this basal oscillation of the nucleus centroid during the time course of the experiment. And so, having this assay in hand, she could then test for the usual suspects that are normally involved in nucleus positioning. So she could test for the involvement of microtubules, and she could see that microtubules have a minor contribution to nucleus positioning. Namely, you can get rid of microtubule, and the nucleus is still able to find its way to the center. More surprisingly, she could show that myosin 2 is not involved at all in the movement of nucleus to the center. And so then she wanted to determine whether potential actin flows or actin polymerization gradients could be involved in mediating this movement. So here I must remind you what we know from the nucleation of this actin microfilament in this model system. So we know from the work that has been mostly done in the group of Melina Shu in Cambridge that the nucleator, both formin 2 and spire 1 and 2, which work in conjunction, are hooked to vesicles. So these vesicles are actually RAB11A positive, such that the microfilaments, they actually grow from vesicles. And we can actually detect these vesicles by labeling the actin filaments with low doses of this F-actin probe, which is a eutrophin coupled to GFP. And you can see that these actin-positive vesicles appear here as black dots, and from these vesicles emanate the filaments. And so Maria could follow the motion of this actin-positive vesicle while the nucleus will be brought back to the center. And she could not detect any obvious actin flows, really not like the one that maybe some of you have seen in cells which migrate and use a lamellipodia. She could not also detect any obvious density gradient of actin-positive vesicles, but what she could see was that it seems that the actin-positive vesicles undergo cortical attraction that is close, in a region close to the cortex, about 10 microns away from the cortex. Their directionality is biased towards the cortex compared to the center, and their velocity tend to increase. So it seems that locally, close to the cortex, these actin-positive vesicles are being attracted by the cortex. And so if Maria plots the squared velocity of this actin-positive vesicle as a function of the distance to the oocyte center. You can see all these dots in red. You can see that it seems that their velocity actually is highest close to the cortex compared to the center. And so when Maria was doing this experiment, we were already collaborating with two biophysicists, Nirgov and Raphaël Voiturier, and by looking at our experiment, they proposed to make an analogy between these actin-positive vesicles and particles in a fluid that are subjected to thermal agitation. So namely, we could make an analogy between these particles and say that their thermal agitation, which is proportional to their squared velocity, is highest close to the cortex compared to the center, such that these particles exert a kind of gradient that is highest close to the cortex compared to the center of the oocyte, their thermal agitation being highest and hence the pressure being highest close to the cortex, such that the velocity of the nucleus is subjected to a propulsion force exerted by a pressure gradient due to the agitation of these actin-positive vesicles. But what Maria also observed is that not only does she have this gradient of pressure exerted by actin-positive vesicles, but also when she looked at steady state in a control oocyte, she could see that all the vesicles from the cytoplasm, so actin-positive ones, but also other kinds of vesicles, were subjected to a kind of a flux. She could actually measure this flux spatially using sticks analysis. And what you can appreciate here is that it's not like this nice flux that has been described in Drosophila. It's more like a cytoplasmic activity that involves the whole cytoplasm. But it's not like a stream. You don't have a unique directionality. It's moving in all directions, more like a washing machine. And you can actually quantify this cytoplasmic activity by performing correlation analysis over time, namely taking region and seeing how the pixel intensity varies with time. And the more the pixel intensity varies, the more rapid the correlation coefficient will drop. So the higher the cytoplasmic activity, the more rapid you will lose your correlation coefficient. And if you compare this situation with what is happening in the formin 2 knockout oocyte, you see that it seems that in the formin 2 knockout oocyte, it's almost as if the cytoplasm is dead. And you can actually see that by sticks analysis, but also measure that by correlation analysis. You see that the cytoplasmic activity drops very slowly compared to the control. And when she puts back formin 2 into formin 2 knockout oocytes, then she regains the cytoplasmic activity along with the nucleus being centered, going back to the center. And this is shown here. So she has, by sticks analysis, but also by correlation analysis, you see that then the correlation coefficient drops almost like white type. And we know that this global cytoplasmic flux is really due to the presence of microfilaments, because if we depolymerize microfilament with cytochalasin D, so these guys, we then treat them with cytochalasin D, then we completely abolish the cytoplasmic activity. The correlation coefficient drops very slowly. Sorry. It has also been shown by the group of Melina Shu that actually the movement of this actin-positive vesicle is due to myosin 5B, to this molecular motor of myosin 5B. And so we decided to test for the involvement of myosin 5B in this cytoplasmic flux. And so Maria injected oocyte, so this is the control, and this is control oocyte injected with a dominant negative form of myosin 5B. And I think you can really appreciate that in this movie, all the vesicles from the cytoplasm are put in motion, whereas here, only the region of the nucleus seems to be moving, but the rest of the cytoplasm seems to be a bit dormant. And actually, this is measured here. If you compare the sticks correlation and the sticks analysis, but also the evolution of the correlation coefficient, it drops much more slowly when myosin 5B activity is impaired. And so how do we interpret this? So we think that the actin-positive vesicles are being moved along microfilaments thanks to myosin 5B activity, and while they move, they are able to indirectly put the fluid around them in motion. So, such as the movement of all vesicles, so not just the cytoplasmic, the actin-positive vesicle, but all vesicles from the cytoplasm are put in motion thanks to the activity of myosin 5B. And so this motion is actually quite important because if then Maria redoes her essay where she puts formin 2 back into formin 2 knockout oocyte, but then she adds this dominant negative form of myosin 5B, you see that instead of allowing the centration of the nucleus and putting back the cytoplasm in motion, then she makes like a kind of jelly. This can be actually seen on this movie, but it can also be quantified here. You see that in the control, so this is this red curve, so the nucleus centroid will migrate from the periphery to the center, and at the same time, if you look at the evolution of the correlation coefficient in red, it drops all the time, very rapidly, whereas when you express the myosin 5B dominant negative construct, so at the beginning, probably the dominant negative hasn't taken its effect, so the nucleus starts migrating to the center, the correlation coefficient drops very rapidly, and then suddenly, the nucleus centroid stops its motion towards the center. It actually goes backwards, goes back to the periphery. And this correlates with the fact that the correlation coefficient of the cytoplasm now drops much more slowly. So suggesting that actually this global cytoplasmic flux promoted by myosin 5B activity help the motion of the nucleus from the periphery to the center. So how could it work? So we decided to directly measure the parameters of elasticity and viscosity inside the oocyte, and to do that, we collaborated with Willy Ahmed and Timo Bess at the Curie Institute, and we used optical tweezers to actually probe the oocyte cytoplasm. And what Maria could observe is that when she overexpressed myosin 5B, then she tremendously increased the elasticity and the viscosity of the oocyte cytoplasm. So suggesting that this motion of all the cytoplasm is actually fluidizing is actually inducing the fluidization of the cytoplasm. So we had to revisit our model. So the nucleus is subjected to a gradient of pressure and a propulsion force exerted by the actin-positive vesicle. And we actually think that this gradient of pressure is non-specific because if we inject latex beads coupled to fluorochrome, these beads are able to become centered, provided that they are sufficiently big to be able to see this pressure gradient. But the nucleus is also subjected to a counteracting force, a friction force, that is due to the viscosity of the cytoplasm, so which is in the opposite direction, and that is proportional to the viscosity of the cytoplasm, such that the velocity of the nucleus is proportional to the pressure gradient of the actin-positive vesicles, but inversely proportional to the viscosity of the cytoplasm. And so, myosin 5B has a dual role. It permits the generation of a non-specific pressure gradient that triggers the centering, but it also indirectly generates a cytoplasmic activity that allows the fluidization of the cytoplasm, which makes it compatible for the movement of such a large object as the oocyte nucleus. So what Maria also analyzed was actually the behavior of these actin-positive vesicles. So, namely, she looked at the tracks of these actin-positive vesicles, and she measured the mean square displacement of these actin-positive vesicles, which is a measure of the space explored by unit of time by these vesicles. And surprisingly, what she could observe is that actually the mean square displacement of these actin-positive vesicles follows a linear regression with a slope of 1 as a function of time. And this kind of behavior is typically the behavior of a diffusive object. This is the behavior of a diffusive object when it's in a medium that is purely viscous. But it has been proposed by Clifford Brongwin and David Weiss that actually in a viscoelastic environment, such as the oocyte cytoplasm as we have measured using our optical tweezers, this kind of motion, which is propelled by molecular motor, which looks like diffusion, could be defined as active diffusion. Namely, it looks like diffusion, but it's happening at faster scale. So, in conclusion, we have discovered a novel mode of actin-dependent nucleus positioning, which relies on a non-specific pressure gradient. This is the first demonstration that a molecular motor is able to fluidize the cytoplasm of a cell. And also, this is the first identification for a cellular function for active diffusion. And so, in my lab, we have studied two very opposite chromosome motion. One motion that I've just described to you now, which is the centering of the nucleus, which happens in prophase 1 of meiosis, and just the opposite movement, which is the off-centering of the spindle and the chromosome that happens in meiosis 1, and which is required for the asymmetry of the division and very important for the physiology of the female gamete. What is interesting is that these two motions, which are opposite, depend on exactly the same nucleators, namely formin 2 and spire 1 and 2, but seems to be using different mechanisms. So one motion actually is relatively slow. It takes 4 to 6 hours, and the speed of the nucleus will decrease as the nucleus reaches the center, whereas the opposite motion is more rapid. It takes 2 to 3 hours, and the speed of the spindle increases while it reaches the cortex. This motion, as I've just showed, is myosin 2 independent. This one requires myosin 2. This motion, I didn't show you this, but this motion is totally independent from the property of the cortex, whereas this opposite motion really requires cortex softening in order to be efficient. And so we would like in the future to understand how this is feasible, how you can reuse the same nucleators, but allow completely opposite motion and give different properties to the mesh. But also another aspect that we would like to understand, which is a bit less trivial, is why do mammalian oocytes bother put their nuclei to their center just before they are actually going like 1 hour later to put them off center in order to allow this extremely asymmetric division that permits the oocyte to preserve its maternal store. And one area of research that we are now initiating, is that maybe this movement of the nucleus from the periphery to the center, which normally takes place in vivo at the end of oogenesis, is important to modulate maternal transcription, because it correlates with actually this very important event, which is the switching off of maternal transcription, which is extremely necessary for zygotic transcription to take place later on after fertilization. So with this, I'd like to thank the people who did the work. So I mentioned, of course, Maria. I mentioned the name of a previous member of the lab, but I would like also to thank all the members of my lab. I would like to thank Philippe Mailly from the STIRB Imaging Facility for his help with the analysis of the mean square displacement of actin-positive vesicles, our collaborator in Curie, William Hen, Mathias Bussionnier, and Timo Belz, and of course our two favorite modelizers, Nirgov and Raphaël Voitourier, and you for your attention.